The title of this talk is an overview of the nervous system histology. This talk will be conducted using a conversational format. Here are the class expected outcome. You do not need to read them. We will use them to chart our talk. The drawing we will use to guide the initial section of this talk is based on a drawing published in 1984 in the Illustrated Encyclopedia of Human Histology, and many years later reproduced with some modifications in Not 3, the Human Brain book. Both are great books to have for those interested in the subject of neuroanatomy. The drawing in this frame is based on those previous drawings. The drawing looks complicated, but it will become clear to you as we introduce each component one at a time. But before we do this, we will give you an overview of the structures depicted in this figure. The larger circle represents the central nervous system. Here I am representing the skin with myelinated axons connected to an encapsulated receptor and unmyelinated axons. Here, the muscle with an efferent myelinated axon and an afferent myelinated axon. This structure represents the bowels. These three elements represent autonomic motor neurons in an autonomic ganglia. Their unmyelinated axons are covered by non-myelin producing Schwann cells, whereas the preganglionic axons going to the ganglion cells are covered by thin myelinated layers. These four elements that I am now indicating represent neurons in the dorsal root ganglia and cranial nerve sensory ganglia. Only the proximal extension of the skin myelinated fiber is represented in this drawing. So now that you have an overview of this drawing, I will start the individual description of each of the cells depicted in it. The first cell I like to introduce is the protoplasmatic astrocyte. They are represented guarding the periphery and in the parenchyma, which you can see at the center of the image I have just introduced. These astrocytes encircle the central nervous system, shielding it from the peripheral nervous system as well as from all other systems in the body, including from vascular structures inside the central nervous system. Notice the shape of the protoplasmatic astrocyte indicated by the arrow and its extension. These extensions are resting on a vessel, as you can see. We should think of them as protectors. They are present in the gray matter. Protoplasmatic astrocytes offer mechanical support, metabolic support to neurons, and in addition, they respond to injury, leading to the formation of scars. Next, we have the fibrous or fibrillary astrocytes. They live in the white matter. They do have a fibrillary appearance, as you can see captured in this picture and schematically here being pointed by the arrow. They serve as mechanical and metabolic support in the white matter and they react to injury. Next, we have the oligodendrocyte. They are represented here in green. They are mainly in the white matter. Oligodendrocytes have a fried egg appearance on histological preparations, as you can see. They are in charge of producing myelin in the central nervous system. Unlike in the peripheral nervous system where every unmyelinated axon is enveloped by a Schwann cell, the equivalent of oligodendrocyte in the peripheral nervous system, in the central nervous system, unmyelinated axons are not covered by oligodendrocytes. Next, we will talk about ependymal cells. They line the ventricles. They are called ependymocytes. In this frame, you can see them forming a fence around these structures. Notice in this enlarged section the little hairs swimming in the CSF. Ependymal cells line the choroid plexus and secrete CSF. This type of ependymal cell is called choroid plexus epithelial cells. The third type of ependymal cell samples the CSF content and informs the parenchymal neurons of its status. They are called tanycytes. Next, I will tell you a few words about microglia. The function of microglia in health is unknown. In case of injury to the central nervous system, they become macrophages, devouring pathogens and debris. You can see in this specimen a bunch of microglia devouring a cell. Here they are producing the cell by phagocytosis. Some scientists believe microglia derive from monocyte-related stem cells that invade the central nervous system early in development. Others that microglia derive from neural epithelium. Satellite cells are present in cranial and peripheral sensory ganglia. They function as protectors, support, and source of sustenance to the ganglion cells. Satellite cells can be considered a modified Schwann cell. You can see them in this frame surrounding multiple dorsal spinal ganglion and cranial nerve sensory ganglion cells, neurons. Satellite cells are also found in the autonomic ganglia, as you see here. Schwann cells are present around myelinated axons and non-myelinated axons, as previously mentioned. They provide metabolic support for both axon, myelinated and unmyelinated axons. In addition, they provide electrical insulation for myelinated axons. The photo shown in this frame corresponds to Schwann cells. Especially noticeable is their elongated nuclei. The arrow in this drawing points to one neuron. This is one of the original drawing of a neuron made by Ramón y Cajal. This frame shows a picture of Santiago Ramón y Cajal, the initiator of the neuronal doctrine when young. This is a picture of him many years later. Yet in both pictures you see that he's close to the microscope. It was the use of the microscope and his intuition that led to our current understanding of neuronal function. Neurons have two functions that are not shared by other cells. One relates to information. In this domain, neurons are involved in collection, and transmission of information. The second neuronal function not shared with other cells is the long-distance transportation of macromolecules. Neurons, in this example, have a multitude of connections, depicted here as pink elements on top of the red structure. They include axon somatic synapse, axon dendritic synapse, and axonic synapse, the latter implying when one axon contacts with another axon. Were we to strip the neuron from all these synapses, then we would see the body of the neuron, referred to as soma or pericardium, and the dendrites with the spines, which are site of axonal dendritic connections, the axon, and the terminal segment, also known as synaptic terminals. The third term used for this region is telodendrites, a poor choice for labeling of the distal segment of an axon. The axon is covered by myelin. In this case, myelin in the CNS is produced by oligodendroglia cells. Now we can look at the neuron and remove a segment of the soma to see what is inside of it. And what we find are multiple organs, including nucleus with its double membrane as seen by electromicropores to allow nucleocytoplasm communication, and DNA-rich matrix. The nucleolus, which unlike the nucleus is rich in RNA. We would also see mitochondria. Mitochondria produce energy for cell metabolism. We would also see mitochondria. Mitochondria produce energy for cell metabolism. Their dysfunction often leads to multifocal strokes, as indicated by the aqua arrow in the MRI, and high lactic acid, the hallmark of mitochondrial dysfunction. We would see smooth endothelial reticulum, an organelle with smooth walls and multiple functions, including the assembly of lipids. We would also see free ribosomes, viewed as a single dot in electron microscopy, or forming polyribosomes. Polyribosome is a gathering of multiple ribosomes, and ribosomes sometimes attached to endothelial reticulum, forming endothelial reticulum. The rough endothelial reticulum in neurons is often called Nissl substance. In the protoplasm, we would also find Golgi apparatus. The Golgi apparatus is characterized by double-layer membrane with closely packed vesicles. One of the functions of the Golgi apparatus, perhaps the most important one, is to attach carbohydrate that function as addresses to guide cellular respective destination. We also find lysosomes. Their function is to degrade complex structures, including glycoaminoglycans, formerly known as mucopolysaccharides. Lysosomal dysfunction produces Hurler disease, characterized among other things by coarse fissures, cloudy corneas, mental retardation, and abnormal looking. These vertebrae are called fish mouth vertebrae. In addition, we have in the neuron peroxisomes. These organelles are in charge of catabolizing multiple substances, including very long-chain fatty acids. Their dysfunction leads to a condition called Zellweger. Another important neuronal structure are cytoskeletal elements. In this category, we have microfilaments made of actin. They are very numerous in the growth cones. Neurofilaments, the main skeletal component of neurons, and microtubules. Myelin is not a component of the neuron, but it is closely linked to it. In the central nervous system, myelin is produced by oligodendroglia. One oligodendroglia producing myelin for segments of axons of multiple neurons. In the peripheral nervous system, myelin is produced by Schwann cells. One Schwann cell only wraps one segment of one axon. Myelin is made of whorls of Schwann cell membrane attached to the body of the Schwann cell by a mesoaxon with fingers of persisting cytoplasm trapped between the layers of the Schwann cell membranes. These fingers are called Schmidt-Lintermann incisures. The arrow is indicating to the axon hillock. The axon hillock is devoid of ribosomes and of Golgi apparatus, but it is rich in sodium voltage-gated channels, making it an important site for information transmission on account of being the most likely place to generate an action potential. This action potential generates an electrical field that in normal circumstances will be carried electrotonically to the next notoranvier and excited to the point of producing another action potential, and then another and another, ultimately reaching the terminal segment and culminating in the release of neurotransmitter. To accomplish this feat, remember that axons may be as long as 1 meter. Transportation of multiple elements to maintain the structural integrity of the axon and often transport the necessary element to sustain transmission. The neuron needs of a resistance axon and a robust transport system in that axon. Here I have removed the top half of the axon cylinder with the adjoining myelin in all the length of the axon to view. I have now taken a segment and enlarged it, as you can see in this frame. I have done this to show you the main components of an axon. These components are mitochondria, microfilaments, neurofilaments, and microtubules. The latter three structures can be readily seen using electron microscopy, as I will show you in the next few frames. This is the same picture we saw when talking about myelin. I am showing it to you again to point out the cytoskeletal elements present in the axon. These relative compact structures are the microfilaments. They are a component of the cytoskeletal system of the cell, as we have previously mentioned. Neurofilaments appear less well organized. They too constitute a component of the cytoskeletal system. The microtubules are the third component of the neuron cytoskeletal system. They indeed have a tubular appearance, as you can see. They are made of tubulins and transporting proteins, capable of moving large chunks of meat by simply modifying their shape, as I am representing in these frames. The protein for fast transport towards the positive pole of tubulin is kinesin. Kinesin in the axon transport elements towards the axon terminal. The protein for fast transport from the axon terminal to the neuronal body is dynein. In axons, as you can see, the positive pole of microtubules is towards the terminal. Tubulins and related proteins are also in charge of fast transport in the dendrites. In the dendrites, microtubules are oriented in both directions. Other elements present in the axons are transport vesicles, lysosomes, and a smooth endothelial reticulum. Most authors I have read do not think that rough endothelial reticulum or free ribosomes are present in axons, but others believe that they are present in a small amount. Axons also have slow transport system, but much less is known about them. So at this point, I will not go further into this discussion. Neurons can be classified according to size. Some are small as 4 microns. Others are big as 125 microns. Granule cells in the cerebellum are very small, and tedial-horn motor neurons are very big. Neurons can also be classified according to axonal length. Golgi 1 have long axons, Golgi 2 short axons. Neurons that form the cortex that projects to the spinal cord are Golgi 1. Those that connect neurons within a hemisphere are called Golgi 2. Neurons can also be classified according to the number of neurites. By neurites I mean axons, and then they are classified as when they have many dendrites and one axon. Multipolar neurons are found in central nervous system and autonomic ganglia, but not in dorsal ganglia. In addition to multipolar neurons, we have bipolar neurons. Bipolar neurons have one neurite at each pole of the cell. These neurons are found in the olfactory nerve, retina, cochlear nucleus, vestibular nucleus, and cerebral cortex. The third group of neurons, classified based on the number of neurites, are the unipolar or pseudo-unipolar neurons. The later term being more appropriate because developmentally they start as bipolar and progressively their neurites to the point of fusion of the process close to the soma. Both processes behave like axons. In these neurons, we see a single neurite that divides into processes. Pseudo-unipolar neurons are found in the dorsal ganglia and also in cranial nerve sensory ganglia. Now I will finish this section by showing you the three neurons every medical student should be able to identify on light microscopy: bed cells of the cerebral motor cortex, dorsal root ganglion neurons, and cerebellar Purkinje cells. The last point I want to make is that when responding to injury, protoplasmatic and fibrous astrocytes change shape. Their cytoplasm increases and become eosinophilic, and the nucleus is displaced laterally. They are then called gemistocytic astrocytes. Thank you very much for your attention.